Good afternoon. Welcome to the annual McNair Conference at Texas A&M University, Corpus Christi. My name is Katherine Lanoue. I've been working for the past year as a McNair Scholar in the Marine Genomics Laboratory at Texas A&M University, Corpus Christi. I've been working on my McNair project under the supervision of my mentor, Dave Portnoy, with the assistance of his lab manager, Elizabeth Hunt, and his postdoctoral students, Andrew Fields and Amanda Barker. My research is taking a look at alternative mating behaviors of the marine catfish, Eriopsis felis, or better known as the hardhead catfish. This species of catfish is most known for their venomous barbs and abundance in the southern Gulf. Additionally, these fish engage in an untraditional mating strategy called mouthbrooding. During their mating season, which takes place between the months of April to June, female marine catfish will release unfertilized eggs into the water near a male catfish that they wish to fertilize their eggs. The male catfish will take these eggs and after fertilizing them, will carry the embryos in their mouth until after they hatch. Mouthbrooding provides additional protection for the embryos until maturation, allowing a higher rate of survival compared to non-mouthbrooding fish species. Though mouthbrooding is advantageous for hardheads as a species, it is taxing for the male that is tasked with carrying the embryos. The male catfish carries the embryos for approximately 7 weeks until after they hatch. It is hypothesized that males carrying embryos may only be able to spawn once per year, and in carrying the offspring, they must expend extra energy and are unable to eat while rearing the young. As such, it would be more beneficial to males to somehow get other males to rear their own young. This brings me to my next point of brood parasitism. Brood parasitism is the laying of eggs in another organism's nest, referred to as the host, with the intention of making the host raise the parasitic young, essentially having the host carrying on another organism's gene line. The practice of brood parasitism is actually seen across taxa in birds, such as cuckoos and cowbirds, amphibians, and fish, including cichlids and cuckoo catfish. It is thought that some of these species have actually evolved special traits to assist in brood parasitism, such as having parasitic eggs look similar to host eggs, or having a lack in sexual dimorphism within a species. My work hypothesizes that multiple parents may be contributing genetic material to a clutch of hardhead catfish embryos through polygyny, or multiple mothers, or polyandry, multiple fathers, by potentially using brood parasitism. Though brood parasitism has never been seen in this species of fish, as a mouthbrooding species there is definitely an incentive to perform brood parasitism. Previous studies have found that there is no significant sexual dimorphism between sexually mature males and females, both being similar in size and appearance. This would potentially allow young, physically immature, hard-headed catfish males, called sneaker males, to come into the nests of other larger males under the guise of being a female fish coming to add eggs to the host male's nest, only to implant their own fertilized eggs or to fertilize any of the other male's unfertilized eggs. There are obviously many advantages to being a sneaker male and having another male catfish carry their young. They are guaranteed that their lineage will continue, but will not have to expend any energy to do so. And without a brood of embryos to carry for months, the sneaker males are then able to mate with multiple females and to commit brood parasitism on multiple host males in one mating season. In order to explore my research questions, I obtained samples of 5 male catfish, each carrying a brood of approximately 20 embryos in their mouths. The DNA of the males and embryos was extracted and sequenced using double digestive restriction site associated DNA sequencing, or ddRAD sequencing. Using ddRAD sequencing, a next-generation sequencing technique, I will be able to determine parentage de novo. This method has also been found to be more robust and efficient than previous methods used for parentage analysis, such as microsatellite analysis. After sequencing, I am going to assemble a reference genome of the species and use this reference to compare SNPs, or single nucleotide polymorphisms, between the parents and offspring. A SNP is a single nucleotide substitution at a specific position or loci on the genome. By comparing specific SNP sites between the carrying males and their offspring, I can deduce if the offspring were sired by a different male or by the carrying male, and by one or multiple mothers. If the offspring are sired by their carrying male, they will have an allele at the loci that matches that of the carrying male, and an additional allele belonging to their mother. However, if there is an allele that does not match that of the carrying male, I can assume that they were sired by a sneaker male instead. Additionally, if there are differing mother alleles, I can assume that the carrying male took part in polygyny. Please refer to Figure 4 on my poster for a graphical representation of potential interpretations of my analysis. Though I do not have my results yet, I do expect to see proof of multiple parents within a brood. I expect that both male and female catfish will mate with multiple partners in order to increase their individual fitness. Additionally, I expect there will be a higher rate of polygyny than polyandry within the broods due to the protective nature of male catfish over their territories, thus providing less opportunity for cuckoldry to occur. Though these fish are not typically caught for sport or as food, they play an important role as a bottom feeder in their environment. Understanding the mating strategies used by this fish can be used in fisheries and stock assessment, and can further our understanding of their development and importance in their ecological niche. Please refer to my poster and graphics for further details regarding the process of sample preparation for ddRAD sequencing and the interpretation of potential results. Please scan the QR code located at the bottom of the poster for additional information about the work and research being done by the Marine Genomics Laboratory at Texas A&M University, Corpus Christi. I want to thank the McNair Scholars Program and Texas A&M University, Corpus Christi for funding my research. Thank you for your time and attention. Have a blessed rest of your day.